Hello, ladies and gentlemen. First of all, I would like to thank the conference organizers for the opportunity to present. I'll be talking about work we conducted over the last years where we have been looking into more complex inheritance paradigms for the inherited cardiac disorders. For those unfamiliar with these disorders are associated with risk of sudden cardiac death at young age. They have been classically divided into two categories. On the one hand, we have the primary electrical disorders, such as long QT syndrome and catecholaminergic polymorphic ventricular tachycardia. In the primary electrical disorders, the heart is structurally normal and the abnormality lies in the electrical functioning of the heart. And these disorders have been linked to rare pathogenic variants in genes encoding ion channels. On the other side, we have the cardiomyopathies, where an abnormal cardiac structure plays an important role in the genesis of arrhythmias causing sudden cardiac death. Among the more common forms of cardiomyopathies are hypertrophic cardiomyopathy, caused by rare pathogenic variants in genes encoding components of the cardiac sarcomere, and arrhythmogenic cardiomyopathy, which is linked to pathogenic variants in genes encoding components of the desmosome. While the monogenic inheritance paradigm allowed us to discover many genes underlying these disorders, several observations suggest that the underlying genetic architecture, at least in some patients, may be more complex than we previously thought. Clearly, monogenic forms of these disorders exist, and in this case, one rare large effect genetic defect by itself determines susceptibility to the disease. Such truly monogenic disorders typically present with a severe phenotype at young age, for example, as observed in the calmodulopathies. On the other hand, the reduced penetrance and variable disease severity that is often observed in families segregating rare pathogenic variants in the established genes points to the presence of other co-inherited genetic factors, typically referred to as genetic modifiers, that act to modulate the effect of the primary genetic defect. Such near-monogenic inheritance is typically observed, for example, in genotype-positive families with the long QT syndrome or hypertrophic cardiomyopathy. Furthermore, some patients remain genotype negative despite comprehensive genetic testing of the known genes. These cases typically present as sporadic cases or with a less pronounced familial clustering of the disorder. In these cases, a more complex inheritance, oligogenic or polygenic, is suspected, where one would need to accumulate multiple risk variants to reach the threshold for the disease. Examples of such disorders are the Brugada syndrome and dilated cardiomyopathy. We expect that variants that have a different frequency and effect size in the germ population underlie such complex inheritance, with both common variants of small effect as well as less common variants of larger effect playing a contributory role. Till now, research has largely focused on the identification of the contributory role of common variants, and this will be the focus of my talk today. Identification of common susceptibility variants has relied mainly on two different approaches. One of them is shown here. This is the case-controlled genome-wide association study design, where common variants in the form of single nucleotide polymorphism spread throughout the genome are directly tested for a role in disease risk. In this design, a greater prevalence of a particular variant in cases as compared to ethnically matched controls would point to a role of that variant in disease susceptibility. Another approach is the intermediate trait approach, or the endophenotype approach, where one first identifies those common variants that modulate traits that represent biological pathways known to be involved in the disease. And once those modulatory variants have been identified, these are then tested for their possible role in modulating disease susceptibility or disease severity in patients. Now, such traits include ECG parameters such as the QT interval, which are of relevance to the electrical disorders, and cardiac magnetic resonance traits of cardiac structure and function that are of relevance to the cardiomyopathies. This latter approach, offers the important advantage that these intermediate phenotypes are amenable to accurate measurement in very large sections of the general population, and we can therefore achieve a remarkably large statistical power for the identification of modulatory variants. The first genome-wide association study on this class of disorders was conducted by Philippe Charon, François Cambien and co-workers in 2011 on dilated cardiomyopathy. And in that study, they had identified two common susceptibility variants for DCM. In 2013, our group applied GWAS for the first time to Brugada syndrome, identifying three common susceptibility variants. Since then, we and others have studied several disorders using this approach, identifying multiple common susceptibility variants for these disorders. Today I shall focus on our recent work on long QT syndrome and hypertrophic cardiomyopathy, which are essentially among the more common types of inherited cardiac disorders. I shall start with long QT syndrome. It is a disorder that typically manifests since childhood and adolescence. It presents with prolonged QT interval on the ECG and is associated with a particular ventricular arrhythmia referred to as torsade de pointe. And this arrhythmia may deteriorate into ventricular fibrillation and lead to sudden cardiac death. About 80% of patients harbor a pathogenic variant in one of the three major ion channel genes, namely KCNQ1, KCNH2, or SCN5A. In order to address the role of common genetic variation in long QT syndrome disease risk, we established the Long QT Syndrome Genetic Consortium that allowed us to bring together about 1,200 unrelated individuals, mostly probands of European ancestry, and 418 individuals of Japanese ancestry. And 11% of these were genotype negative, meaning that comprehensive testing of the known long QT syndrome genes did not uncover a rare pathogenic variant. We conducted two case-control genome-wide association studies in the European patients and in the Japanese patients, respectively, comparing each to ethnically matched controls. What you see here is a Manhattan plot emerging from the meta-analysis of those two genome-wide association studies. Essentially, we identified three common variant susceptibility loci at genome-wide statistical significance, for the first time providing strong evidence for a role of common variants in disease susceptibility in long QT syndrome. And not surprisingly, these loci had all been associated with QT interval duration in the general population, with QT interval being a very important intermediate phenotype of the long QT syndrome. We explored further the genetic overlap between risk for long QT syndrome and genetic control of the QT interval in the general population by conducting genome-wide correlation analysis using our long QT GWAS data and previously published QT interval GWAS data obtained in the general population. We demonstrated the genome-wide genetic correlation between genetic risk for long QT syndrome and genetic control of QT interval in the general population. In contrast, as expected, we found no correlation with other ECG parameters such as heart rate, PR interval, and QRS duration. This essentially points to shared genetic factors between these two phenotypes, long QT syndrome risk on the one hand and QT interval duration in the general population on the other hand, which essentially provides empirical support for the utility of the intermediate phenotype approach for the identification of risk variants. Genetic overlap between long QT syndrome risk and genetic control of the QT interval in the general population is also supported by the analysis shown here. We here related the effect size of 68 SNPs that were previously associated with QT interval in the germ population at genome-wide statistical significance with the risk that these confer for long QT syndrome. This uncovered a strong correlation between the two effects, further supporting the presence of sure genetic determinants and rationalizing the intermediate rate approach for the discovery of modulatory genetic factors. In line with their high frequency in the germ population, common genetic variants are almost invariably associated with a small effect size. For this reason, in an effort to account for a greater portion of disease susceptibility or disease variability, we consider their effect in aggregate in the form of a polygenic risk score. In its simplest form, a polygenic risk score calculation comprises counting the number of risk alleles of an individual. Yet we know that not all SNPs are created equal. Some SNPs carry a larger effect on risk as compared to others, and in an effort to account for this, we weight each risk allele by its effect size to generate a weighted polygenic risk score, which is deemed to be more accurate. Using the 68 QT SNPs, we calculated a weighted polygenic risk score and compared its distribution in the long QT syndrome patients and controls. And this demonstrated, as expected, that patients with long QT syndrome had a distribution that was shifted to higher polygenic risk scores. But the more interesting observation emerged when we then distinguished between genotype-positive individuals, that is, those that had a rare pathogenic variant in KCNQ1, KCNH2, or SCN5A, and genotype-negative long QT syndrome cases. This showed that polygenic risk score distribution was shifted to higher scores in genotype-negative patients compared to genotype-positive ones, suggesting that unlike the genotype-positive patients, where the effect of the rare variant predominates, genotype-negative patients have a more polygenic architecture wherein common variant burden plays a more prominent role. And this is important as it has implications both for gene discovery efforts as well as for genetic counseling of patients. So that was all about disease susceptibility, but what about effects of the polygenic risk score on disease severity? Now this was rather disappointing. We found no association between the polygenic risk score and QT interval among the patients that we studied. Similarly, we found no association between the polygenic risk score and time to life-threatening arrhythmic events, as you can see here in an analysis stratified by polygenic risk score quartiles. Now, this was at variance with two other previous studies, including this one from our group, where we did find a relation between a polygenic risk score based on 22 QT SNPs and disease severity among patients with a KCNH2 genetic defect. An important difference, however, between this study and the current study is that this one included both probands and genotype-positive relatives, whereas the GWAS study included only unrelated individuals, mostly probands. We believe that the inclusion of both probands and relatives results in greater variation in QTC and is thus expected to increase statistical power for detection of modulatory effects. Another possibility could be that while in the study shown here, all patients harbored a mutation in the same gene, that is KCNH2, in the current GWAS study we included patients with different rare variants in different genes. And these may be associated with biophysical defects of varying severity, which are also expected to contribute to inter-individual variability, which is difficult to account for in the association analysis. The study from the Ackerman group also included both probands and relatives and found a higher mean polygenic risk score in patients with a high QTC, that is QTC above 480 milliseconds, compared to those with a QTC below 480. This study also found a higher polygenic risk score in probands who we know are typically more severely affected versus family members. On the other hand, this study found no association between the polygenic risk score and occurrence of symptoms. So while studies so far have found strong support for the role of common variants in determining susceptibility to long QT syndrome, more work is needed to understand the role of common variants in modulating disease severity. We believe that findings so far call for an evaluation of QT SNPs in larger sets of patients comprising both probands and their genotype positive relatives, and these studies are ongoing in the setting of the EJPRD funded project Long QT Syndrome Next. We hope to be able to include enough patients to also allow us to analyze the different major genetic subtypes, that is long QT1, type 2 as well as type 3 separately, or to adequately account for possible effects mediated by mutation in the different genes. We are currently including patients in the study and would love to hear from you in case you would like to participate. Clearly, as more QT SNPs are identified in the GAM population by means of larger GWAS on the QT interval, we can incorporate these into more comprehensive polygenic risk scores and possibly be able to explain a larger part of inter-individual variability in disease susceptibility in the future. I now shift to hypertrophic cardiomyopathy, which affects 1 in 500 individuals and is a leading cause of sudden death and heart failure in early and mid-adult life. About half of cases with hypertrophic cardiomyopathy harbor a rare pathogenic variant in genes encoding proteins of the cardiac sarcomere. In affected families with an identified genetic defect in these genes, similar to long QT syndrome, incomplete penetrance and variable disease expression is often encountered. Outstandingly, about half of patients remain genotype negative after genetic testing, and in these patients, the genetic causes have remained unknown. Earlier this year, 2 large collaborative studies, one of which led by our group and another one led by Professor Hugh Watkins in Oxford, reported on effects of common genetic variants in HCM disease susceptibility and severity with comparable results. In our study, we conducted a Genome-wide association study comparing 1,733 cases with HCM recruited in the Netherlands, the UK and Canada to ancestry-matched controls, identifying 6 susceptibility loci. Among these loci was a locus on chromosome 18, overlapping the FHOD3 gene, which had been previously uncovered in a smaller HCM GWAS published in 2013. We then noticed that this locus had also been previously associated with dilated cardiomyopathy, but interestingly with an opposite direction of effect. This is evidenced by comparing the odds ratios for the 2 associations, highlighted here in yellow. These odds ratios are expressed for the same allele. We see an odds ratio above 1 for the association with HCM, whereas we see an odds ratio below 1 for the association with DCM. So in fact, we saw that the risk allele for HCM actually constituted the protective allele for dilated cardiomyopathy. This observation was not limited to one locus. We also noted the same for the locus on chromosome 10 located in the BAG3 gene. Similarly, this locus was previously associated with dilated cardiomyopathy with an opposite direction of effect. That is, the HCM risk allele is protective for dilated cardiomyopathy. Furthermore, through FEWAS in the UK Biobank, we saw that this locus was associated with left ventricular ejection fraction, where the hypertrophic cardiomyopathy risk allele was associated with an increase in left ventricular ejection fraction. So essentially, a picture started to emerge where HCM and DCM shared at least some risk loci, and that these loci had an opposite direction of effect. Furthermore, there was an indication that these loci may affect risk through effects on cardiac contractility, with left ventricular ejection fraction being a volumetric measure of cardiac contractility. So this inspired us to conduct a series of additional genome-wide association studies to investigate further the genetic relationship between hypertrophic cardiomyopathy, dilated cardiomyopathy, and cardiac contractility. Why is this important? While clinically increased contractility, as typically evidenced by an increased left ventricular ejection fraction, may form part of the clinical picture of HCM, we do not know whether this is actually causally related to HCM, and by conducting these genetic studies, we can address that question. In collaboration with colleagues at Imperial College London, we conducted GWAS in individuals from the UK Biobank on 9 different left ventricular traits derived from cardiac magnetic resonance imaging. These are summarized, are listed over here and comprise left ventricular volume, left ventricular thickening, as well as left ventricular contractility traits, all plausible intermediate traits of hypertrophic cardiomyopathy. This is a combined Manhattan plot displaying the lowest p-value of each SNP for any of the 9 left ventricular traits on which we did the GWAS, and in all we identified across these traits 17 genome-wide significant loci. We also conducted a meta-analysis of 3 previously published genome-wide association studies of DCM, identifying 3 genome-wide significant loci. We explored the genetic correlations between each of the 9 LV traits, HCM and DCM, by means of LD score regression. Pairwise genetic correlation between the 9 LV traits and HCM are shown here in red, whereas the correlations with DCM are shown in blue. HCM showed a positive genetic correlation as expected with mean wall thickness, while DCM was positively correlated with left ventricular and diastolic and end systolic volumes. Decreased contractility is a hallmark of DCM, and we observed a negative genetic correlation between DCM and LV contractility parameters, whether assessed using a volumetric measure, that is left ventricular ejection fraction, or using global LV strain measured in any direction, that is circumferential, radial, and longitudinal. Of note, all 4 of these contractility parameters were positively correlated with hypertrophic cardiomyopathy. That is, increases in contractility are correlated with increased HCM risk, highlighting a divergent relationship of contractility parameters between HCM and DCM. We went on to demonstrate a causal relationship between increased left ventricular contractility and HCM risk by conducting 2-sample Mendelian randomization where the exposure variables were left ventricular contractility measures and the outcome was HCM. We then moved on to exploit the genetic correlation between HCM, DCM, and the 9 LV traits to boost the statistical power of our HCM GWAS. We did this by combining the LV trait GWASs and the DCM GWAS with the HCM GWAS in a multi-trait analysis of GWAS approach, MTAG. For HCM, this took us from the 6 loci that we previously had to 16 HCM-associated loci in the MTAG analysis. These 16 loci are tabulated here. In the penultimate column, you can see the association p-value emerging from the MTAG GWAS for HCM. We looked up that association p-value in the original GWAS, and you can see this in the 3rd column from the right. For almost all of the loci that emerged in the MTAG analysis, we see that they had exhibited an association p-value that was just below genome-wide significance in the original GWAS, almost completely all of them, underscoring the likely validity of these loci. We also had the opportunity to validate these loci in an independent set of 2,694 HCM cases from the sister study of Hugh Watkins. These comprise patients from the HCMR registry and the National Institute for Health Research Bioresource for Rare Disease Patients Resource. And 15 of the 16 HCM loci were replicated in this analysis. We looked at the direction and strength of effect of the 16 HCM risk alleles across all 9 LV traits and DCM. This is represented as a heat map over here, and as you can see, many of the HCM risk alleles are associated with reduced risk of DCM, and in the general population with increased left ventricular contractility and decreased LV volumes, in line with the divergent relation between genetic risk of HCM and DCM. We then proceeded to test these genome-wide significant SNPs in the form of a polygenic risk score for an association with disease severity in HCM. This was done in an independent set of 368 HCM probands and their relatives, all with a mutation in a sarcomeric gene. This uncovered an association of the polygenic risk score with maximal left ventricular wall thickness, where each standard deviation in polygenic risk score was associated with a clinically relevant 1.4 mm increase in maximal left ventricular wall thickness. Furthermore, we found an association with the occurrence of adverse clinical events composed of a composite of septal reduction therapy, cardiac transplantation, sustained ventricular arrhythmias, sudden cardiac death, ICD therapy, and atrial fibrillation. Here you see event-free survival in sarcomeric mutation carriers stratified by polygenic risk score above versus below the median. Now, while this data is promising, clearly, besides further validation of our observations to date, as mentioned for lung QT syndrome, future clinical utility of such a polygenic risk score in HCM relies on our ability to increase its discriminative capacity by incorporating additional modulatory SNPs. So, in summary, it is clear that common genetic variants unequivocally play a role in modulating susceptibility to inherited cardiac disorders. Furthermore, evidence is accumulating that common variants modulate disease severity in patients with rare pathogenic variants. Also, we've shown that the study of intermediate traits is an important means of identifying the contributory common genetic variants and can help us identify biological pathways that are involved in disease pathogenesis. And the question now remains, the open question that now remains, is as to whether we will be able to identify sufficient SNPs in order to allow us to generate polygenic risk scores that are discriminative enough for clinical utility. I would like to end by thanking the collaborators on the studies that I have presented today, who are clearly many. Most of all, I would like to thank two very talented young scientists who spearheaded this work while they were in my group. I would like to thank Rafik Tadros, who conducted the work in the hypertrophic cardiomyopathy studies, and Najim Larouchi, who conducted the work on long QT syndrome. And finally, I would like to thank you for your attention. Thank you.